In this video, we're going to talk about proteins. So it's another class of biomolecules. In fact, protein comprises about 20 to 30% of your mass of your cells. Most of these proteins have pretty varied functions. It's because they can fold and be arranged in a variety of different ways. What we find is that proteins have the most sort of functional variety of all biomolecules. So structurally, they can help keep your cell in a certain position or shape. They're also involved with motility and movement. Chemically, proteins can be used for enzymes, and they're also involved with contraction of both cells and muscles. So they play an important role with things like cell division and even muscle cell contraction. So like other biomolecules, it also contains carbon, hydrogen, oxygen. Nitrogen is an important component of protein, and sometimes sulfur and phosphorus. Now, proteins are actually polymers of what we call amino acids. And these amino acids are the monomers of protein that are all held together by peptide bonds when forming a polymer. Now, the shape and function of the protein itself is at 4 different structural levels. Now, all proteins are made from 20 different types of amino acids, and these amino acids are joined by covalent bonds, which we call peptide bonds. So when you have lots of amino acids in a long chain forming a polymer, those amino acids are held together by a special type of bond called a peptide bond, which is made of a covalent bond, but it's a specific structure of a bond that I'll explain here in a minute. Now, these peptide bonds contain an amine group and a carboxylic acid group, and when they join together, that's what contributes to the peptide bond, and they can act either as an acid or as a base. So these amino acids can be acidic or basic, and they differ because there's 20 different amino acids that have these different R groups. Think of R as being like the rest of the amino acid, and each R group is different, which gives the 20 different amino acids a separate structure. So this is showing the basic anatomy of an amino acid. You're going to have an amine group on one side, which is nitrogen and 2 hydrogen atoms. You're going to have a carboxylic acid group on the other side, which is a carbon doubly bound to an oxygen and a hydroxide group, as well as a central carbon atom with a hydrogen atom off the side, and then an R group over here. Remember, R can be just a different molecule of some sort. R can be as simple as a hydrogen atom or as more complex as something like a double ring structure. So R is what makes each amino acid different. Otherwise, the amino acids are identical in terms of this structure here. R is the part that can vary. So what we see here are 2 different amino acids. Remember, amino acids are the monomers of a protein. So 2 monomers can come together through dehydration synthesis to make a dipeptide. Remember, peptide bonds are what hold amino acids together. So you see the peptide bond is actually where the amine side of one amino acid links up with the carboxylic acid side of another amino acid to form this special type of bond, which is called a peptide bond, which is pretty stable. Also, these peptide bonds can twist. So these amino acids can spin around each other around this peptide bond, which gives them some flexibility and foldability as well. Now, dipeptides can also be broken down into their individual monomers or amino acids through hydrolysis, just like other biomolecules. So in terms of the structural levels of protein, we got 4 major levels. The primary level is just a linear sequence of amino acids. So when you talk about placing amino acids in a particular order as a polymer, that's the primary sequence there. Now, the secondary level of structure is how primary amino acids interact within each other. They can form things like alpha helices, which are coils that can resemble a spring, or even beta pleated sheets, which resemble kind of an accordion ribbon. Now, the tertiary level is basically where secondary structures interact within each other. So you might have like folded beta sheets or alpha helices that can twist upon each other. And the quaternary level is basically how 2 or more different polypeptides or proteins interact with each other. An example of quaternary is things like hemoglobin, where there's 4 separate proteins that all interact with each other to make one large protein called hemoglobin. So this is showing the primary structure of a protein. So you can see that you have each individual amino acids that are all linked up by peptide bonds. You see here the peptide bonds, and these link up these amino acids into a polymer. This is just showing the basic primary structure of amino acids. Now, what makes proteins different from each other is the fact that the sequence of amino acids can be different. And because we have different amino acids, which can be placed in different sequences, it gives this primary level some flexibility in terms of having varied structure. So what's interesting is that the primary structure of a protein often determines what its secondary structure is. So that based on the sequence of amino acids, it may coil into an alpha helix, or it may fold on itself into a beta pleated sheet. And this kind of forms like a zigzag. Now it turns out these are more stable forms of protein, and these, depending on their structure, also determine the function of the protein. So these alpha helices might be involved as like a receptor site on a particular membrane protein, or these beta sheets might serve as a structural protein in a tissue of your body. So just different examples of another structure of protein. Now the tertiary level of structure is where you have superimposed secondary structure, where you have alpha helices that might kind of fold on themselves or have beta sheets that kind of interact within itself too. And so it can form a more complex protein that could be globular, and it's held together by intramolecular bonds. But this can be something like an enzyme, or it could be another protein that could serve a structural role, or it could serve as a receptor in a cell. It can even serve as like a gene regulator. These are very complex types of proteins that have a variety of functions. But what's important to note about these proteins is that their function is determined by their structure. So if you change the structure of a protein, you're also going to alter its function. So that's important to regulate protein structure to keep sort of a constant function inside of your cells. Now the quaternary level is basically where two or more polypeptide chains interact upon themselves. So what we find is that this might be two separate tertiary proteins that are actually interacting with each other to form one large quaternary level of protein. And an example of this could be something like hemoglobin inside of red blood cells, which is involved with the transportation of gases. Now, their proteins can be either fibrous or globular, and so a fibrous protein is typically structural. It's going to be strand-like, water-insoluble, but also very stable. Most of these are tertiary quaternary structures, and they provide mechanical support and tensile strength to your cells and tissues. Some examples of fibrous proteins can be things like keratin, which is what makes up your hair and nails and gives skin some strength. Elastin and collagen are also very abundant fibrous proteins you find in skin that help to give skin some structure and resiliency. And different contractile fibers are also a different type of fibrous protein. Now, globular proteins are more compact and spherical, and they are water soluble, so they're more likely to dissolve within the watery solutions of your body. So some globular proteins could be things like blood proteins. These are very sensitive to environmental changes, though. So if you have a change in pH or temperature, it can alter the shape of this globular protein, which can also alter its function. Like the fibrous proteins, they're at the tertiary and quaternary level of structure, and they have active sites, which are the functional regions, and these can serve as things like antibodies for immune defense, hormones for cell signaling, or molecular chaperones for transportation of different molecules or even atoms and ions. And globular proteins can also function as enzymes, which can speed up chemical reactions. So one thing that's important to note is that there's this thing that can occur called denaturation. And denaturation is basically where globular proteins unfold and they lose their three-dimensional shape. Fibrous proteins are more stable, so they're much less likely to denature. But when proteins denature, their active sites also deactivate, and therefore they can lose their function. So denaturation can be caused by a decrease in pH, or like an increase in acidity, or an increase in temperature. They're usually reversible because we actually have enzymes that help to renature or sort of put back together denatured proteins. We call those heat shock proteins. And otherwise, they can be irreversible if the changes are extreme enough. For instance, you can't undo a cooked egg. So the process of cooking an egg denatures the protein of egg, and you can't undo that denatured albumin. Unfortunately, you can't reverse cooked egg in its kind of solid state back into its uncooked or undenatured liquid state. Now, enzymes are often made of protein, and these act as catalysts. So what a catalyst does is it increases the speed of a chemical reaction without actually getting used up in the process. So these enzymes can speed up chemical reactions, but they're reusable. And they speed up chemical reactions by lowering the amount of energy that's needed to start or initiate that reaction. So by lowering that activation energy, this actually leads to an increase in the speed of the reaction. And what's amazing is some enzymes can allow for millions of reactions every minute. But without these enzymes, some of these chemical reactions might take years to occur spontaneously, whereas an enzyme can speed it up on the order of millions per minute instead of one per year. So some of the characteristics of enzymes is that we refer to basically functional enzymes as hollow enzymes, and these here are two parts. We have apo and cofactors. Apo enzymes are a protein portion. The cofactor is the metal ion. Remember, a lot of iron and copper and zinc are often cofactors for enzymes, and coenzymes are going to be things like organic molecules like vitamins that help also with the structure of an enzyme. Now, enzymes are specific, which means they only act on a very specific type of molecule, which we call substrate. So substrates are those things that are acted upon by enzymes. Usually the name of an enzyme ends in ase. So if you see that the name of a molecule is something like hydrolase or oxidase, that ase tells you that it's going to be an enzyme of some sort. Now, not all enzymes are made of protein, but most are. Now, enzymes act by lowering the activation energy. And really the activation energy is the energy required to initiate a chemical reaction. So enzymes kind of prime the reaction. They get it going by lowering that energy required to get it started. And enzymes allow for chemical reactions to proceed very quickly at body temperature. Now, there's 3 steps that are involved in enzyme action. We need a substrate to bind to the enzyme's active site. Remember, active sites are the portion of the globular enzyme or protein where the substrate can bind. This temporarily forms an enzyme-substrate complex, where effectively the enzyme forms a weak interaction with the substrate. What happens then is that the complex undergoes a rearrangement, and so that the enzyme might change shape, and that the substrate also changes shape. And this results in the final product where the product is released from the enzyme, but the enzyme is not used up in the reaction, so we can go back to the first step where the enzyme is ready to bind to another substrate. And this whole circle or process can occur millions of times per minute. So what this is showing is basically how enzymes speed up chemical reactions. And this graph shows the total energy of our system as well as the progress of our chemical reaction. We see that without enzymes, chemical reactions have a large activation energy hump to get over in order to reach their product. This energy barrier makes it that these chemical reactions occur more slowly because it takes longer and takes more energy to get over this barrier to reach your product. Now, with enzymes, effectively those enzymes lower the activation energy, and because the energy barrier is smaller, this reaction occurs much more rapidly, and it's easier to reach your end product. Now, in terms of the steps of enzyme action, remember, you need an enzyme here with your active sites. This is showing a globular type of enzyme. Now, these active sites are what bind to your substrates. Remember, the substrates are the molecules or atoms that are being acted upon. Now, the first step here is that we need to form an enzyme-substrate complex. And what's important to note is that enzymes are specific to substrates. Now, once this complex is formed, this enzyme-substrate complex undergoes what we call a change in shape or a conformational change, where energy is used and this bond is formed here. So you might actually be, linking these particular monomers together. And this could be an example of like dehydration synthesis. And now the third step is where you have the enzyme and product that dissociate, and now you have a new product here, where we had 2 different substrates that are now joined together. And this is showing an example of how a dipeptide can be formed by an enzyme. Now this enzyme is not used up in the reaction, and so this can actually go back and really just serve the same function as it did before. And this can occur so quickly, it can occur on the order of millions of times every minute.